Hi, my name is V, and I'm a rising high school senior interning at the Kamenetska Lab at Boston University. Deoxyribonucleic acid, or what most of us know as just simply DNA, is one of the four main molecules of life. DNA molecule consists of two strands that wind around each other to form a double helix, a structure that is commonly associated with the DNA molecule. Each strand consists of nitrogenous bases, thymine, adenine, cytosine, and guanine, that pair and link the two strands together. The function of DNA is important to all living organisms because it carries the genetic information that we need to grow, function, and reproduce. It has been suggested that DNA conductance is involved with several biological processes. Researchers have attempted to measure the conductance of DNA by attaching linkers to the end of the strands that would then bind with gold junctions. However, the results of these experiments have been inconsistent and sometimes contradictory. We believe that the main problem to this is that the question of how DNA binds to metal junctions is still unclear. Here, my project in the Kamenetska Lab focuses on the conductance of two guanine analogs. We chose these molecules because of its ability to easily dissolve in water, as well as its resemblance to the structure of guanine. We run the solution in a scanning tunneling microscope, or STM for short, which is an instrument used to image surfaces at the atomic level. Parts of the STM include a gold sample and a gold tip. We use a technique known as the break junction technique, and we repeatedly bring the gold tip into contact with the sample and then withdraw. The gap that is formed is narrow enough to be bridged by the molecule, and then the current traveling through the junction is then measured. We find that the guanine analogs can conduct and do not need a linker to bind to the gold junctions. We hypothesize that the guanine analogs are binding through the native nitrogens. The next step in our research is to work directly with DNA bases. Studying DNA conductance and how they bind is important if we wish to implement DNA in electronic devices as well as to recognize damage in the molecule.